7, 9, 10. Okay? 7, 9, and 10, right?
Yeah.
Okay, 2/3, anterior 2/3 is cranial nerve number 7. Posterior third is 9. And then back here, where you see these big old guys, that's 10. Okay, so that's all first order. Okay, name the cranial nerves that control the anterior 2/3, or this area of the tongue. I won't have to say 2/3 of the tongue.
Is it facial?
It is facial. Okay, cranial nerve number 7. First order, second order, third order. First order.
Oh, yeah.
Yeah. Okay, goes to medulla. And it goes back. Okay, the primary gustatory area is the insula. Okay, so the insula means island. So you open this up like this. Wah, wah, wah. Right? And there is that little island of brain tissue. So that's where we're going to realize gustation. Cool? All right, and then the taste. We're not going to do the histology, but you know we have the valate, the eights in front, and the forms in the back. Cool. Okay, so and since we can't, I can't get one. I can just say, hey, name a taste bud that's back here, and you're going to put form and then something in front of it. Fungiform or what's the other one? Filiform. Okay. Cool. All right. So that's those two. Then we do the eye. And we do its neurology. Okay, so we have the optic nerves coming through here. We don't have a good chiasmatic sulcus, okay. But we do have a crossing, so it's going to go from here to the, where are we at? The lateral geniculate, okay. So this is the lateral geniculate right here, number 27. Cool?
The bended knee, right?
Hmm?
It's the bended knee.
Exactly, but it's the lateral bended knee, not the medial bended knee. Cool?
27?
27. Right here. Okay, remember 27 and 28 go together because a superior colliculus is a visual reflex, right? So you have to get that to the superior colliculus so that when someone comes whizzing at you over here and you happen to, you know, it's like. Did you ever see that on The Matrix when he's like. Superior colliculus, right? And it's like a rubber back. How he did that was pretty cool, huh? I think it was special effects.
This is that
Okay. And then the optic tract goes back, so I can't, there's nothing to tag here unless, no, see, we come from here to there, and then we'd have these radiations that go back this way, which we don't have any, so can't tag that, so basically. And then, obviously back here is vision in the occipital lobe. Okay, so what was the big picture on vision?
Light passed through the retina.
Hmm?
The light passes in the 3D effect, right?
Well, yeah, but
Light waves.
what's the big picture and everything? We got to convert some kind of energy
Yeah, you
to
light-
light
light
into
nerve
a
energy.
signal.
So we converted odorants to a signal so that we can tell what we're smelling. We converted some tastants to a signal so that we can understand what we're what we're eating. So this we have to convert light energy to pulses so that we can interpret what we see. Okay, so we have to concentrate the light in. So all these structures here are basically for that purpose. Cool? So we started here. We got the upper lid and the lower lid, and we talked about a conjunctiva, and there it is. This is the palpebral conjunctiva. But we also talked about these little blue and 7 and 8, these little plate in here. Those are the tarsus plate with the meibomian glands in it. Okay, and I'm not going to test you on it, but we said if they get clogged up, they make a pathology. Hmm?
Shall I say?
Yeah.
Yeah, she can say it the best, so refer to her. I can't say it either. Chaz Leon. I always call it Charlene Theon. Okay, and then we come in. I'm getting really good at this. I'm doing a portrait right now, and I've been drawing this lady's eyes for days now because I keep getting it wrong. But I'm saying, man, that canthus is just too far out. So I had to erase the eye and draw it back in. But I use the medial canthus as my marker. Okay, so medial canthus, lateral canthus.
Could you tell us the numbers? So we can write down
Something
Medial canthus doesn't have a number. It has a, oh, well, I'll tell you what. You're asking these worn out eyes to look at numbers that small? Okay, so medial canthus is just the inner corner of the eye. Lateral canthus is the outer corner of the eye. Okay, just remember corner of the eye.
Is that little
Okay.
pink thing next to the medial canthus, is that the caruncle?
Caruncle.
Caruncle. Okay.
Yeah, so that's next on the list. Okay, so we have tarsus gland, which you have to take off. Okay, then we have the medial canthus and the lateral canthus, and then we have the little pink thing in there, which is the caruncle. The conjunctiva we really can't see. I mean, there's the palpebral conjunctivus, and then the bulbar conjunctivus would be on the eyelid ball itself. And remember, it folds on itself. So we have this membrane, comes here, it folds up, goes to the upper lid. From the top lid, it folds on, into the upper lid.
So that pink is the palpebra.
Yeah, palpebrae, because palpebrae means eyelids.
Okay.
Okay, bulbar is referring to the bulbous eye.
There's two separate ones.
Yeah. And of course, you can say, okay, he probably won't ask that because he's got nothing to tag. Okay, which is true. All right, there's our Charlize Theron and Sty. Okay, it sounds like a singing group, right? Okay, so the eyelashes just have the sebaceous oil glands in them. The eyebrows, okay, don't have eyebrows. I'll have to get an ex-student, let them stand there and tag their eyebrow. Okay, but again, that's to control the glare. Everything around your eye basically is to keep, you know, so you can close your eyes and squint if there's too much light, stuff like that. All right, we have to keep the eyes moist and wet. Okay, this is the lacrimal apparatus, right? Lacrimal glands are always lateral, so we know that this is a left eye. We know that this is a right eye because of this. Cool? All right. So the lacrimal glands here, lacrimal gland is controlled by cranial nerve number what?
3.
Not 3.
Lacrimal. Oh, so 5.
No, that's fission.
7.
4?
7.
Oh, dang it.
No,
That was
what are
4.
okay? What are your parasympathetic the cranial nerves for the parasympathetic system? 3, 7, 9, 10. Okay, 3, 7, 9, 10. Make sure you get that. 37, 9, 10. Cool. Okay, 3 is only for like accommodation and pupillary control. Okay, 7 is crying and spitting. 9 is spitting. 10 is everything else. Every visceral function you have. Okay, 9 is also for swallowing. Cool? Okay, so lacrimal gland here. Then we have the little tubes. They're all green. Okay, those are your ducts. Those are your lacrimal excretory ducts. So we produce the product in here, and we excrete it into the ducts, and then the ducts go over the eyelid, over the eyebrow, over the eye, and wash the eye from lateral to medial. Okay, and superior lateral to, well, it goes to both sides. I was going to say inferior medial, but it doesn't. It goes up and down. Okay, so it washes the eye this way, which is a good idea because if you just have your lacrimal gland here, then your outer side's going to get all dried out. Not good. You can't keep your contact lenses on that way. Cool. Okay, so your lacrimal puncta is. Does yours have a puncta on there?
Yes.
Yeah, so you see the little black dots in there. Okay, those are actually openings, okay, which you can see on yourself. Just pull down and look at them, okay? they lead to your ducts, to the superior lacrimal canal. Okay, so superior and inferior. I mean, that's just directional terminology. Okay, and then they all meet here in the lacrimal sac. The nasolacrimal duct. Are you really videotaping that? I just screwed that up. Can I
I'll
do
edit
like
it.
in a,
It's fine.
can we do a cut and then?
I'm gonna turn it into a song.
Okay. And there we have it. Okay, so it goes down in.
Lacrimal duct is
This is the sac, so just the top of it, and then the rest of it is the duct,
okay.
going from up here into the nose.
Okay.
Good? All right, so while we're here, we have the eye muscles. Okay, we learned this last semester in 24.1. Superior rectus, inferior rectus, medial rectus, lateral rectus, superior oblique, and both models only have a tendon. Okay, so make sure you understand this is the tendon to superior oblique. And then they both have inferior oblique. This is inferior oblique here, and there is the inferior oblique on that one. Okay, so you see how it's attached right there at the lacrimal bone. Okay, because there's the lacrimal canal. Cool? All right, so what are their motions? What does superior rectus do?
Down.
Up. Superior rectus, not oblique. Okay, medial rectus, in. Lateral rectus,
Out.
out. Okay, inferior rectus, down. Okay, superior oblique, down and in. Inferior oblique,
Out.
up
and
and up, in and up.
out.
Okay,
In
they're both here. One's up, one's down. Cool. Okay, and so conjugate eye movement. Okay, so I can put a question, one eye or the other, and say, okay, name this muscle, right? You'd say lateral rectus. I said, what's its conjugate? Medial rectus. Okay, so you'd have to say, name this muscle. You have to put which one it is. Left lateral rectus. What's its conjugate? Right medial rectus. Okay, superior oblique. What's its conjugate? Inferior oblique. This is rectus. Remember, if this is a rectus, this is an oblique. If this is an oblique, this is a rectus. Okay, and it's opposite. So what's the conjugate for this one? Well, first off, name this muscle.
Superior.
Superior oblique. What's its conjugate?
Inferior rectus.
Inferior rectus. Yeah. Okay, because we both want to look down. Cool. This one's going to look down and out basically because remember they're at this angle right here. They're not straight back, so they're at this angle here. So this one's actually going to actually torque it just a bit. Cool. All right. So we talked about conjugate movement. Okay. What are the nerves?
3, 4, 6.
No. What are the nerves that control? Well, yeah, 3, 4, 6. But which does? I tell you what. What does 4 control?
Inward movement.
No. What is it? What nerve? What does cranial nerve 4? What muscle does it control in the eye movement? You what?
Lateral rectus.
No, it's superior oblique.
Superior, yeah.
4. Okay, so what you're going to do for this is you're going to study 4 and 6. Okay, so what does 6 control? Do what? Lateral rectus. Okay, so these two, 4 and 6. So if I tag anything else and I say, okay, name the muscle, name its conjugate, what nerve controls it? If it's not this or that, it's 3. Okay? 3 does everything except these 2. So if you learn this is 4, superior oblique, this is 6, then you have that part of the test covered. There's no way I can get you, right? Not that I'm trying to get you anyway. I want you to know this stuff. Cool? All right, so let's look at these things. I'm sort of going the way I'm going to lay out the test. Okay. Those of you that haven't before know that I lay it out exactly the way I keep it. Okay. All right. So sclera, cornea. Obviously, the cornea is clear. Why is the sclera white and fibrous?
It's thick. It's for.
Yeah, because it's protection. Anyone want to voluntarily poke them in the eye?
No.
I didn't think so. But what do you think would happen if you get poked in the eye?
If we got poked in the eye?
Not with a sharp stick, but just with your finger. I mean, if you didn't have this,
Oh, it goes
any
straight in
poking
there.
in your eye would puncture it, right? Okay, yeah, a sharp stick is going to, but even then, that sharp stick is going to have a little bit of force because you can't just wink, wink, wink. It won't happen. This thing is that tough. All right? That's it. Fibrous we're talking tunics, okay? So one layer, second layer, last layer, or third layer. Okay, so that's it for that layer. So the vascular tunic is the choroid layer. Okay, so it's this. So you can see it here. And then here's the dissection of it, showing you graphically what's in there. So that's your nerve supply. I mean, that's your vascular supply. Provides nutrients to the eyeball. Okay, basically it provides nutrients to the retina. Okay, to the rods and cones by diffusion. Cool. Contains melanocytes to prevent light from scattering. So this is the side that has the melanocytes in it. Okay, so you got to remember this thing is embedded in that. All right, the ora serrata, remember this thing is circular, like this. Okay, let me see what this says in there. Oh. Yeah, this isn't very good. Okay, so there's your ora serrata too. Okay, so I can tag this if I want to. Okay, so there's the ora serrata coming all the way around. And it's basically the ciliary body. Okay, so here's what it looks like if you cross-section it. Cool? Okay, so the ciliary body has ciliary muscle, 21, which alters the shape of the lens. So it actually, when it's relaxed, it makes the lens, keeps the lens flattened, or stretched. When it contracts, it does like this, and it loosens the slack on these suspensory ligaments and lets the lens get big and fat. Okay, so at rest, long vision, contracted, near vision. Cool? Okay, and there's the suspensory ligaments, and they're all the way around. The lens, well, we don't have the lens here, but the lens is pretty nondescript. I mean, it's made of crystalline protein layers like an onion, okay? The iris is the colored portion, so this is the iris. This is the iris. Look at that iris. Okay. The pupil is the hole, okay? Let's just go back together. So the pupil is the hole, obviously. What's the whole purpose?
To let light in.
To let light in, because we got to convert that energy to a pulse. And then the pupil has muscles. Okay, so we have circular, outer circular, and we have circular muscles that are under parasympathetic control. Okay, so circular muscles are going to do this, almost like the shutter of a camera, and then the radial muscles are going to pull it back out. Okay. How are you going to remember which one's parasympathetic, which one's sympathetic? How are you going to do that? Eyes wide with fright, right? Because when you're in a danger situation, the last thing you want is your pupils to pinpoint, right? They go big. The tarsus plate holds your eyelids open, and you can see the danger, and then you need to get away, or you need to fight. Hence the nickname fight or flight. Cool? All right. All right, so continue on in here. You know, going to be over there. So something from in here is going to be here, and then I move over to the next thing. Okay, so the nervous tunic is the optic disc. Okay, so this is your blind spot. Okay, and notice that it's not straight back, that it's actually at an angle. Remember, it's going to be an angle like a 15-degree angle. All right. Don't like me, man. I don't care. I don't like you either. Okay, so it's at an angle like this, so it comes back this way. Okay, so this is where that's located. That's why it's a blind spot. Okay, it's going to be about right there, and there are no rods and cones. No rods and cones there. So you can't see anything if there's no rods and cones. So that is your blind spot. Cool? This, however, is the fovea centralis. Okay, where are we? The macula lutea is like 34 on this thing, and then 35 shows a pinpoint in there. So that is the fovea centralis. Okay. So think of it. Okay, so we got this bullseye that has the highest concentration, and then right in the middle is the very, very highest
It's
concentration.
like the sharpest vision, correct?
Right, and that's why you want to see that. Now, you ever noticed yourself cocking your head for your dominant eye? Well, if you ever notice anyone cocking their head to try to see better, that's because, first off, it's their dominant eye. That's why they cock their head that way. Okay, so they cock their head, but they want to get that light right there. So that's the reason someone does that. Okay, now you guys are all so young with these great eyes. Me, however,
I can't see far at all.
I gotta
Like
do
their
that.
whole head to the dominant eye, like this.
Well, you know, you can tell your dominant eye. You do like this, and let's see. You find that. I forgot how he did that. There's a way that the optometrists do that. I just had an eye exam, but you can always like get something and you put it here and you line it up. Your thumb. They said that my left eye is my dominant eye, but actually it's my right. Cause see, this stays right underneath it, and you know it's right underneath it, right? See, it goes off to the left whenever I use my left eye. So it's my right eye is actually my dominant eye. They keep telling me it's my left eye. I'm going like, so
Maybe
why
your
do I
brain
cock my head
thinks.
this way?
Maybe your brain thinks it's your better eye.
Could be these contact lenses. I just can tell you when I was younger and playing basketball all the time, I would do like this to shoot the ball, okay? And I was guaranteed to make it, okay, because I was good. I can tell you anything. I can't prove it anymore, right? All right, so this is the retina. Okay, this is back here. Remember, light goes this way, so it says the light rays. So this 43 is actually this little rim right here. Okay, so this is your little
Retina.
melanocytes coming in here. Okay, so these are your layers of rods and cones. Then we come where it breaks. Remember, these are bipolar neurons. Okay, so when you come here, so 40 is rods and cones. Then we have 39. Okay, 39 is going to be the bipolar cell layer. Okay, so one layer, two layers, so two layers of bipolar cells. Then these are cell bodies. Okay, can you see that? Those are the cell bodies of the optic nerve. That all these things are going to coalesce and become the optic nerve. So this is your ganglion cell layer.
38 between this section or right here?
38 is the ganglion cell layer. 37 is the optic nerve. Okay, so we got layers of rods and cones, and they're showing you rods and cones. Then we have the bipolar cell layer. Then we have the ganglion cell layer. Then we have
The optic nerve.
the axons of the bipolar. I mean, of the ganglion cell becomes the optic nerve. Then that's what goes to the lateral geniculate, and stops there. Cool. All right. Of course, we talked about. I won't be testing you on pathology. Pathology is, well, maybe I can throw a question in there. It'll be a bonus if I throw it in there. All right. So now we move on, like over there, another one of these models, and we're talking about the anterior cavity, which is from here on up. Okay. So we divide anterior cavity and posterior cavity. We already talked about the ciliary process. Posterior chamber, anterior chamber. Okay, remember, chambers. We have cavities, and then we have the chambers within the cavities. So make sure you keep that in mind. Cool. There's your sclerovenous sinus right there. Okay, right there. See that opening? Right there. Don't be afraid. You can look. It's right there. I mean, if you really want to see it up close and personal, do like this. Cool. Okay, so this is how one of the reasons glaucoma happens, because if that gets clogged up, or you don't start draining the aqueous humor that's in there, then you get pressure in your eye. And that's what they do when you put your chin and they. Okay, we don't have a vitreous body, and I don't even know if we have any bullseyes or not. We can dissect one on Wednesday, and I can show you that stuff. I won't tag it, but it's good. It's good to see it. Okay, so macular degeneration or detached retina. Some of that, because it's a big old jelly-looking thing. I mean, in this eye it would be like this big, and it really looks. It looks kind of like. Do you know what chutney is?
No.
Well, it looks like jelly. I mean, the consistency of jelly, so that it really is, and it comes out. It's clear, obviously, because the light has to go through it. But that's what holds this thing back there. So you got a detached retina. Part of that is because that vitreous body is wearing out. It's drying up. And things just tend to dry up as we get older. Not fun, huh? All right. Now we do the hearing. So I love this one. Would you grab that one, Paula? Is it Paula?
Pauline.
Pauline, right? My mother was Pauline or Paula, depending on if you were my cousin or you were my mother. All right, so we're moving on. We'll be coming over here. We do all the eye stuff, so then it morphs into the hearing. All right, where's my brain?
You send that video to me, please.
Okay.
She got everything.
She has the review.
I'm still filming. I always try to get a good view. I'm still filming right now.
Oh, really?
Don't laugh at my whispering comments.
All right, so, okay, so what's the big picture on this one?
Sound waves hit the eardrum.
Right, so we have to
tickles
get
the hair cilia.
Right, exactly. So we have to do mechanical energy, sound waves, because that's mechanical energy, and we have to convert that to an electrical signal and take that to the brain. Okay, so sound waves are going to get converted. Okay, so the vestibular cochlear is going to be somewhere in here. Where is it? Is it labeled here? It's actually going to be one of these right in here. Okay, vestibular cochlear in here. Cool. Okay, so vestibular cochlear nuclei and the ipsilateral medulla oblongata. So this is your medulla oblongata. Cool. Okay, so we're going to go through this one. We're going to synapse here in the medulla oblongata. We're actually going to go bilaterally, and we're going to process the hearing in both hemispheres. All right. This is the olive. Okay, so that's your second order right here. This is the olive. This is the olive. Where is Katya? What number is that?
Which one?
Right there.
37.
37. Okay. How do you say that in Russian?
37.
37?
37.
37. Okay. It's almost like 37.
Yeah.
316.
37.
37.
316. Like,
Yeah.
Like 315?
30 is 30. Same is 7.
Oh, same.
Same.
Did you say him?
Same.
Sem.
Like
100.
7, same.
100.
37.
Yes.
Okay, so there's the olive, and then we're going to go to behind it. Okay, so there is the inferior colliculus, 29. 20, what number?
9.
29. Okay. Actually, this is backwards. It should go medial geniculate, 25?
I think it's either 5 or 6.
It's 26.
26 and 29, 27, 28. Remember, lateral geniculate goes with superior colliculus. Medial geniculate goes with inferior colliculus. Okay, because we want to put hearing to hearing. Okay, so the medial geniculus is all part of the thalamus. So remember, it's still going to the thalamus. But this particular part of the thalamus is called the geniculates. Okay, so inferior geniculate, medial geniculate is hearing, inferior colliculus is hearing. Okay, and you understand how that works? Okay, superior colliculus was you had to, when you did this, okay, you hear this sound. Okay, so you have that sound waves, it goes to the medial geniculate, goes to the inferior colliculus, from the inferior colliculus to the superior colliculus, so you turn your head and find out where did that sound come from. Okay, is that
Which one is medial geniculate?
medial?
Okay.
Remember, if you're medial and lateral, if you have a medial and lateral relationship, you are a geniculate. If you have a superior and inferior relationship, you are a colliculus. Okay, does that make sense? Okay. All right, so there's that. And then the auditory centers in the superior temporal gyrus. Okay, so we're just talking in here. Okay, superior temporal gyrus. All right, now. So we had all these outer structures in the eye to help concentrate, take rid of glare and all that stuff. What is the function of the outer structures of the ear?
To catch the sound.
That's right, to conduct those sound waves into the inner ear so that it can activate the tympanic membrane. Okay, so made it real easy. I mean, there's all sorts of stuff in here. Okay, so we have the helix. The tragus is actually this. Okay. This would be the antitragus, okay, the symbiconcha and all that stuff, but make it simple. This is the lobe, okay, which, I mean, it's basically there to hang something off of it, okay. It really doesn't help conduct, but, you know, what would women do, what would men do if they didn't have a place to hang their earrings, right? Mm-hmm. Cool? All right, then we have the external auditory meatus, which a meatus is just a canal. Okay, so don't be fooled. Let's name this structure. External auditory meatus, because we have an internal auditory meatus too. All right, then we have the tympanic membrane, which is the eardrum itself. Okay, and you can see it's got translucent. Okay, and you can see the bone from this side. So when you are checking ears, can I borrow your? Okay, so you'll get the mechanism. You'll have them tilt over a bit. You'll pull the earlobe down. That's another reason why we have the earlobe. And you'll come in there, and you'll be looking for that cone of light. Okay, because that's going to create a shadow, and you're going to be looking for that. Okay, that's how you can tell that they have a functioning tympanic membrane. That's clear. If there's a perforation in it, you won't see that very well. If it's cloudy for any reason, like it's infected and stuff like that, or it's real bright red, okay, then you see that, okay. What's one of the main things you look for when you look in the ear? Because why can't they hear? What's your first thing?
Drainage.
Do what?
Drainage.
Yeah, is there wax in the ears? Can they not hear because there's too much wax in there? You know, I was having a problem hearing when I was younger, and they put tubes in my ears, and they took my tonsils and adenoids out because that's what they did back then. Okay, but the tubes were to help drain the earwax. I always had, you know, ear infections and all that stuff too when I was little, so that probably explains why I can't hear as well as I should. Cool? All right, then your ossicles. So it goes this way. This is what is attached to the tympanic membrane. Right, there's the cone of light that you're looking at. Okay, so this is the malleus, the hammer, the anvil, and then the stirrup. Okay, but we don't say that. We say Malleus Incus Stapes. Cool? All right. Then we come here, and there is the oval window. And you know why they call it the oval window? Because it's oval. See, that one is round. So how do you tell the difference between a round window and an oval window? One looks oval, and the other one looks round. Okay. So they weren't silly, were they? Oh, we missed or did we get? No, we still have the muscles there. Okay. The ear muscles are this one here. Which one is this? If you only have a choice between tensor and tympani and stapedius. No. Tensor tympani, it goes to the tympanic bone, okay? This, remember, if you're attached to the stapedius, to the stapes, you must be stapedius, right? If you're attached to the tympanic membrane, you must be tensor tympani. Okay, so logic, where it attaches to, that they're not real creative with these these muscles. Okay, if we named them in English, you wouldn't have a problem with it, right? But since they use Latin, then it's like, it's a big word. No, tensor tympani. It tenses the tympanic membrane. Okay, so if you got a lot of sound coming in here, and this thing is just like boom, 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 boom. Well, you want to tamp it down. It's like putting your hand on it, right? And then you don't go deaf. Does that make sense? Mm-hmm. Okay, stapedius. What does stapedius do? It pushes. Remember, sound came into the tympanic membrane. It hit this. It hit that. It caused this to do this into the oval window, right? So if you want to control the hearing, you want to control the loudness coming through here, you can also stop the stapedius from jumping in like this, right? We got a little muscle, stapedius, that helps you do that. Okay, so we got two ways of controlling too much sound coming in there, too much activation, right? Stapedius will slow down the stapes from doing its thing, and the tensor tympani will stop the tympanic membrane from vibrating. Excellent, huh? Most excellent. Cool. All right. Now we're getting to the hearing mechanism itself. Okay, and we're going to have to get this guy. All right, I'll come back again. All right, so we said this is the base. And this is the helicotrema. Okay, so gotcha. What's that number?
12?
12. Is that 12?
I think it's a letter H. Yeah, it's a letter H.
Okay, it's the letter H right there for helicotrema, right? H is for helicotrema, right?
Mm-hmm.
Okay, so, and what I want you to know is that these things are in there. Okay, so if we take this off, you can see this orientation in there. Could you help me out here? Okay, so see, the blue, let's do it this way. The blue is the organ of Corti. Oh, do it this way. We got people. Okay, so the blue is the organ of Corti, and then we have the tympanic scala vestibuli and the scala tympani, scala vestibuli. Did I have it right the first time?
Tympani is the lower one.
Is this one?
Yes.
Okay, you guys get it right. Okay. I can afford to get it wrong, okay? But anyway, because this is the mechanism. Okay, sound's going to come in, and we got fluid. We're going to create a fluid wave when we start pushing the stapes into the oval window, right? And it's going to bounce this thing like this, causing this thing here to bounce on the hair cells, which is going to cause a sound wave. Convert the sound energy to the electrical energy, which is this is your cochlear nerve. Good. All right. So let's see. Where's our organ of Corti? Oh, there we are. Okay. So we have the basilar membrane. I got it now. Okay, so this is your basilar membrane. This is actually the vestibular membrane, and this is your tectorial membrane. Cool? Obviously, okay, so this is where the nerve is, right? The nerve is going to the brain. So this is medial, this is lateral. Okay, so inner hair cells, outer hair cells. Make sense? Okay, so inner hair cells and outer hair cells. And by doing this, okay, so when we're at the base, closest here, this is the base, this is the helicotrema. You notice the size difference? Okay, these are big and these are small. Okay, so actually, that's not the point. These should be short because this is where you're going to excite. This is not done right. Okay. You're going to excite low pitches at the base and high pitches at the helicotrema. Okay, just because slow sound waves can travel further because they're long and loopy. Short sound waves are really short and big, okay, with a lot of high energy. So they're not going to make it past very far from the base. And they're not going to make it to the helicotrema. Okay, so that's why these are going to be bigger at the helicotrema and short at the base. Almost didn't say that right. Okay, so semicircular canals actually goes with they're misplaced here. They shouldn't be here. The bone, that is the bony labyrinth, but this should be with the equilibrium. Okay, so what's the big picture with equilibrium?
Both fluid moves with the direction of your head.
Yeah, so we have to convert the direction of your head to an electrical signal so we can tell which way we're going. Okay, whether we're going straight, up and down, or whether we're rotating in any kind of way. Okay, so that we're taking angular movements and circular movements, converting them to a signal to our brain so that we can understand where we are in space. Cool? And that's our third part. Remember, there's 3 parts to equilibrium, to balance. There's the muscles, the muscle spindles in your muscles and the Golgi tendon organs in your tendons, okay, that are constantly giving you feedback. Also your mechanoreceptors, your Pacinians, Meissners, Ruffinis, all those things are sending signals to the olive, to the cerebellum, to the opposite cerebrums. Okay, we have vision is also giving us that. We can see that we're upright. We can see that we're sitting. We can see how far in the distance. We have 3-dimensional vision, so we know, not
Depth
to just run into
perception,
each one.
yeah.
Yeah. So we have depth perception. Then we have just this. That detects all those things, and oh, I'm falling backwards. I can stand upright. Cool? Okay. Hey, what happened to... Let me see that one. Okay, so there's our orientation, right? This is orientated the same way as this one. Okay, you see that? Okay, so now this makes sense. Anterior canal, posterior canal, horizontal canal. Good? Okay, so if you're going, if you don't have this to orient you, you just know if you're going towards the cochlea, you're anterior, right? Okay, then posterior and horizontal. Good. Okay, so what's the movements? First off, vertical orientation, vestibular nuclei, and the pons and the medulla. So, where's my brain? I'm always leaving my brain somewhere. Okay, so pons and medulla, vestibular nuclei and the pons and the medulla, it sort of covers like in here. Okay, it still has to go through the cerebellum. It goes here, then it goes through the cerebellum before it goes anywhere else. We have third order is ocular motor, trochlear abducens. Okay, and remember, this is the vestibular ocular reflex, right? You can maintain your eye on a target and move your head. Okay, so if you can do that, and you can do it any way you want, you have to have your movers of your eye be connected to this. Does that make sense?
Yeah.
You have to have 3, 4, and 6 connected to the vestibular system, or you can't do this, right? And it always moves opposite the way your head is moving. Okay, so if your head goes down, your eyes go up. If your head goes to the left, your eyes go to the right. Good?
Mm-hmm.
All right. What else do you need to do this? The movers of the neck.
Mm-hmm.
So what controls the movers of the neck?
11.
11. Okay, among others, but 11 is the cranial nerve that controls sternocleidomastoid in the upper trapezium. Then it goes to the thalamus, to the cerebellum, and then we go to the vestibular centers in the parietal lobe. Okay, so just parietal lobe. Cool. That's why one of the functions of the parietal lobe is spatial awareness because of that. Getting this headache from dropping my brain everywhere. Look at this. Oh my gosh. My midbrain, look at it, how it is. My superior colliculus is now my inferior colliculus, and my inferior colliculus, okay. I was panicking there for a minute. All right. So, utricle, saccule. Okay. Utricle's on top. I always go US, US and A. So there's utricle, saccula, ampulla. Okay. So utricle and saccule are for linear movement. Okay. So that means straight forward, up and down. Okay. Utricle and saccule. Okay. There's a macula located in there. We got the stereocilia. Really don't have anything to tag on there, so just the growth structures that we're talking about here. Okay, semicircular canals we already talked about. Anterior, posterior, horizontal. What's the function of this one?
Up and down.
No, these are circular movements.
Oh,
Okay,
circular.
So head movements. Okay, so now look. Make it logical for yourself. This is here. Look at the orientation that it has. This is your ear. So it's like this. So side to side movement,
Oh, it's like the side.
right? Horizontal is easy. No movements. Okay, and then your posterior canal, look how it's oriented. So you're gonna do yes, or back and forth movements. Cool? So not even knowing, I say, okay, what's the movement for this? You don't even need to memorize it. Just look at it. So, oh, well, look how it is. It's oriented, like this. So, boing, boing, boing. Cool? All right. All right, and that's that. Okay, then we go to the endocrine system. Of course, we're going to be like way over here on the endocrine system. Let's do right here. We got a time on that right there. Okay, so the endocrine system is hormones. Okay, so the pineal gland is right here. And the thing about this, the structures are easy, but do you know what they produce? Okay, so that's the most important thing. Okay, so this is the pineal gland. Pink pineal. Mm-hmm. It's pink and pineal. Cool? Okay, so what's it do?
Melatonin.
Melatonin, right. So it's a part of your sleep-wake cycle and stuff like that. We're going to convert serotonin to melatonin and go to sleep. All right. Then we looked at the pituitary gland here. I'm not the pituitary gland. Yeah, the pituitary gland. What am I talking about? Okay, so this is the infundibulum. Okay, this is the neural part of it. This is the adrenal hypophysis. Okay, so that's called pituitary gland the hypophysis. Okay, so this is the neural hypophysis and the adrenal hypophysis. We don't need to get on the microscope to see that. Okay, but we know that this is nervous tissue that shot down, and that this is glandular tissue that came up during embryology from the stoma, and sort of grabbed onto the neural part, and now we have the two parts.
That's anterior, right?
Hmm?
Anterior and posterior.
Okay, anterior, posterior, yes, ma'am.
Oh, that one's posterior? Oh.
Yeah. Okay. I prefer neuro and adeno because it's more descriptive. Okay. And okay, so what hormones are created here? Remember, we got two categories.
Growth hormone.
We got acidophils and basophils, and that's what we're going to look at over there in the microscope. Okay, so the acidophils has like 2 under its category, and then the basophils has like 4 under its category, right? And you just systematically associate what goes with the acidophil. And like I said, on the test, I'll say, okay, what hormone is associated with this cell? You can give me any of them. You can give me the category, the broad category, or the specific one. Okay, so corticotrope, or it can be ADH, or whatever it is. Okay, so that's how that goes. All right, here's a thyroid gland. Pretty easy. Okay, this actually is the insula, or the connecting part, right? And so these are the 2 wings. That's easy. Name this gland. Thyroid gland. Pretty easy. Name that gland. Parathyroid gland. Okay, then name this gland. Name that gland.
Adrenal. Adrenal.
Adrenal, yeah. So if you know that this is, the kidneys are also called the renals, or the Latin for kidney is renal, then okay, adrenal. And I'm not going to use that one. That's part of the pancreas. Okay, so this is the pancreas. Here is the pancreas. Okay, head, body, and then tail. Okay, so the head is embedded in the duodenum, and the tail is embedded in the spleen. And then in between is the body. Okay, so, and remember, this has a dual function, so we'll see it again during digestion. And the gonads we're going to study with reproduction. So we're not doing these. Okay, so pretty simple. The gross anatomy is not hard at all. Okay, so over here. These aren't in the order that we taught, but this is your kidney. I mean, this is